Good day, everyone. Welcome back to General Biology 1 lecture. Again, this is your mom, Jeremy Sagario, your lecturer for this topic. Why is it necessary for cells to reproduce? The ability of organisms to reproduce in kind is a basic characteristic of all living things. In kind means that the offspring of any organism closely assemble their parent or parents. Sexual reproduction requires fertilization or the union of two cells from two individual organisms resulting in a fertilized egg or zygote. Once a being is fully grown, cell reproduction is still necessary to repair or regenerate tissues. For example, a new blood and skin cells are constantly being produced. All multicellular organisms use cell division for growth and the maintenance and repair of cells and tissues. For single-celled organisms, they use cell division as their modes of reproduction. The continuity of life from one cell to another form has its foundation in the reproduction of cells by the way of the cell cycle. The cell cycle is an orderly sequence of events that describes the stages of a cell's life from the division of a single parent cell to the production of new daughter cells. The mechanisms involved in the cell cycle are highly regulated due to the presence of different checkpoints. Generally, cells go through a cycle of alternating stages of division and rest from division. The cell cycle has two major stages. The first one is the interphase, and under interphase, it is again divided into three stages or phases. First, we have the G1 or the gap 1, the S or the synthesis phase, and the third we have the G2 or the gap 2. And these three phases, as I've mentioned a while back, are under the interphase. During interphase, the cell undergoes normal growth processes while also preparing for cell division. In order for a cell to move from interphase into mitotic phase, many internal and external conditions must be met. After G2, we now have Prophase, Metaphase, Anaphase, and Telophase, wherein these four phases are under mitosis. And after telophase, we have now the division of the cytoplasmic contents, which is called the cytokinesis. So, the cells will divide and the cytoplasmic contents are separated. We now call this process as the cell division. So now let's start by identifying the different stages under the cell cycle. First stage or event is the interphase, wherein it is a part of the cell cycle that occurs between divisions. Take note that interphase is a resting phase, meaning to say it is a non-dividing stage which is devoted largely to cell growth, and most actively dividing cells spend some 90% of their time at this stage. And take note, during this event, we have different checkpoints that monitor the cell during its resting period, wherein they play a big role in the avoidance of errors during the process. An interphase is divided into three gaps: the G1 or the first growth phase, the S, the synthesis phase, and the G2 or the second growth phase. So next, let's proceed to the different stages or phases under the interphase stage. The first stage of interphase is called G1 phase or first gap because from a microscopic aspect, little change is visible in this phase, wherein it is characterized by growth and development. This is because during G1, the cell is quite active at the biological level. The cell is accumulating the building blocks of chromosomal DNA and the associated proteins, as well as accumulating sufficient energy reserves to complete the task of replicating each chromosome in the nucleus. There are actually two checkpoints in G1 stage. First, we have the G1 DNA damage checkpoint that can evaluate the DNA's integrity, wherein if the DNA is with irreparable damage, it will not proceed to the next phase. The second one is restriction checkpoint, wherein it evaluates the capability of the cell to undergo cell division. Take note that if the cell is ready, it will proceed to the S phase, the next stage after G1, and if not, the cell goes to gap 0, wherein cells in this gap can be quiescent or dormant cell or senescent, the aging or deteriorating cell. The next stage of interphase is the S phase or the synthesis phase, wherein chromosomes in the nucleus replicate. In here, DNA replication can proceed through the mechanisms that result in the formation of identical pairs of DNA molecules. Also, the centrosome is duplicated during this phase. These two centrosomes will lead or will give to us the occurrence of mitotic spindle fibers that are very essential for the movement of chromosomes during mitosis. Take note that in S phase, we have a checkpoint called sDNA damage checkpoint, wherein this is present in this phase to monitor the replication process. The last stage is called the gap 2 or the second growth phase, wherein the cell rapidly grows and some organelles are duplicated. Protein synthesis continues that is important in the manipulation of chromosomes. There are two checkpoints in this stage. First, we have the G2 DNA damage checkpoint, ensuring the smooth flow of events under the gap 2 stage. And we have unreplicated DNA checkpoint that is responsible in ensuring that DNA synthesis is complete before proceeding to cell division. After interphase, we now have cell division. Cell division is an important life process. It results in the production of new cells. As organisms live, they are in constant state of repair and disrepair. Their cells grow, old, and are damaged. And to survive, organisms must have a means to replace dying or injured cells. There are two common types of cell division that cells may undergo. Mainly, we have mitosis and meiosis. But for this session, we will just discuss mitosis and its stages. So what is mitosis? Mitosis is a cell division wherein parent cell produces two diploid daughter cells. And we have here a representation for mitosis. The parent cell in the diagram contains 46 number of chromosomes, and the produced daughter cells also contain 46 number of chromosomes, meaning to say the parent cell and the daughter cells are identical in nature. So take note, when we are talking about daughter cells, we have two types. One is the diploid, which is the product of mitosis, and it is being represented by a symbol 2n, and the number of chromosomes involved are 46 when we are talking about human beings. The second type of daughter cells is the haploid, wherein haploid daughter cells are the product of meiosis process, the second type of cell division, wherein the involved in meiosis are the sex cells or the gametes. It is being represented by a symbol N, and the number of chromosomes involved or present to the daughter cells are 23. Now, let's discuss the different stages of mitosis. The first stage is called prophase or the first phase, wherein Formation of mitotic apparatus such as spindle fibers are evident. During prophase, the chromosomes condense, wherein the sister chromatids begin to coil more tightly with the aid of condensing proteins, and became visible under a light microscope. Centrioles begin to move to opposite poles of the cell. In their movement, spindle fibers emerge from the centrioles. The mitotic spindle continues to develop as more microtubules assemble and stretch across the length of the former nuclear area. The nuclear envelope starts to dissociate or break down, and nucleolus also disappears. Also, during prophase, spindle fibers will attach to the kinetochore of the chromatids. This happens because each sister chromatid has kinetochore in the centromere region. The proteins of kinetochore attract and bind mitotic spindle microtubule. Take note that spindle microtubules that do not engage in the chromosomes are called the polar microtubules. These microtubules overlap each other midway between the two poles and contribute to the cell elongation. Astral microtubules are located near the poles that aid in spindle orientation and are required for the regulation of mitosis. Now, let us understand the different parts of a chromosome or a sister chromatid. The center part of the chromosome is called the centromere. The centromere is very important as it is involved in connecting the two sister chromatids. Beside the centromere, we have now the kinetochore that is important in the attachment of kinetochore microtubules. Take note that this spindle fiber here is actually called kinetochore microtubule. It is a microtubule that is attached in the kinetochore of a sister chromatid. We actually have three types of microtubule. Kinetochore microtubule that is attached in the kinetochore. We have the polar microtubule, the overlapping microtubule, and we have the astral microtubule, the one that is located at the poles of a certain cell. And inside the sister chromatid, we have here now the DNA and proteins. The second stage of mitosis is the metaphase, or the so-called the change phase. So what is happening during metaphase? So during metaphase, 46 chromosomes, or all the chromosomes involved in the process, are actually aligned at the center of the cell. The center of the cell, which is this part, is called metaphase plate or equatorial plane, wherein it is actually a midway between the two poles of the cells. Aside from that important event, also remember that the sister chromatids that are aligned in the metaphase plate are tightly attached with each other through the involvement of cohesion proteins. And another one is that chromosomes are maximally condensed during this stage. And at the end of metaphase, we have here the result. The next stage of mitosis is called Anaphase, or the upward phase, wherein the most important event in Anaphase is the separation of the sister chromatids. How is it possible that sister chromatids can be separated? During anaphase, the cohesion proteins degrade and the sister chromatids separate at the centromere. Each chromatid, now called a chromosome, is pulled rapidly toward the centrioles to which its microtubule is attached. During the separation of sister chromatids, the cell becomes visibly elongated or in oval shape as the polar microtubules slide against each other at the metaphase plate where they overlapped. So at the end of anaphase, the chromosomes are at the opposite poles of the cell, and we can see that the cell is elongated. The last stage of mitosis is called telophase or the distance phase, wherein spindle fibers disintegrate, chromosomes disperse, and nuclear membrane reform. During telophase, the chromosomes reach the opposite poles and begin to decondense, relaxing into a chromatin configuration. The mitotic spindles are depolymerized into tubulin monomers that will be used to assemble cytoskeletal components for each daughter cell. The nuclear envelopes form around the chromosomes, and the pleurals appear within the nuclear area. So at the end of telophase, we have here an elongated cell with two quantity of nucleus inside. After telophase, the cell now will proceed to cytokinesis or cell motion, wherein it is the second main stage of the mitotic phase, during which cell division is completed via the physical separation of the cytoplasmic components into two daughter cells. Take note that the process of cytokinesis of cells with cell wall is different than those cells without cell wall. In animal cells, contractile ring forms at the equatorial plate. As the ring contracts, it pulls the equator of the cell inward, making it able to form a cleavage furrow or a fissure. As the ring continues to contract, the cleavage furrow deepens, then divides into two daughter cells. For plant cell, the Golgi vesicles come together at the metaphase plate, forming a pragmoplast or a vesicular structure. As more vesicles fuse, the cell plate enlarges until it emerges with the cell walls at the periphery of the cell. At the end of cytokinesis, we have now here two daughter cells that are diploid in nature.